Hi all, welcome back. And the last two tissue types, I'm going to lump them together um, to talk about. And the reason why I'm doing that is because we'll spend time talking about each of these separately in much greater detail. But that's nervous and muscle tissue. And another reason why I lump them together is because they're both what we call excitable tissues. Excitable tissues mean that they can respond to stimuli. Okay, so normally what will happen is a signal or stimuli will be sent, and then that will develop into some kind of change in the system. Okay, and that change could be a chemical change, that change could be a contraction change, so the cells could contract, they could relax. There's lots of ways at which this can happen, but that's why they're kind of called excitable tissues, is because they have the ability to change with stimuli or with a signal. Okay. So when we talk about this excitability or this excitation, we're normally talking about a change in voltage. We're talking about a change on one side of the membrane and the other side of the membrane. Okay, so maybe one side is negative and the other side is positive, and then through stimuli that those charges are flipped or change, which can drive work. Okay, it can drive a signal, which then can excite work. Okay, and we often discuss what's the membrane potential, okay, or what's the voltage difference between one side of the membrane and the other side of the membrane. Okay. When we talk about nerves, and we'll talk a lot more about nerves in later lectures, okay, they change the voltage rapidly, okay, and that allows for the signal to move at a very rapid rate, okay, which we call an action potential, okay, or an action wave. Okay. Muscles, on the other hand, when they react to that change in voltage, they contract. Okay, and so that causes the cells to shorten. And you'll see when we look at muscle fibers are elongated cells. Okay, sometimes they're multinucleated, sometimes they're single nucleated, but they tend to be long, and contraction would mean that the cell shortens and then it rebounds. Okay. All right, so let's look a little bit at nervous tissue just to kind of get a framework of what nervous tissue would look like. Okay, so again, it's communicating by both electrical and chemical signals, okay? And it consists of what we call a neuron. That's the nerve cell. And that nerve cell has the ability to detect stimuli, respond quickly, and transport some kind of signal back. Okay. In this case, it's a signal back to where the stimuli is coming from, or it's a signal to a region of the body that needs to respond to the stimuli. Okay. And we'll talk about different ways at which this happens. Okay. Outside of the neuron are small cells that often are considered helpers or housekeepers of that neuron. Okay, and those are called neuroglia cells or just glia cells. Okay, and the the usefulness and kind of the idea of glial cells is, I don't want to say it's up for debate, but we're still discovering what's going on because there has been some reports that some glial cells can actually change into neurons. Okay, so these would act like stem cells in a way, and they would mature into neurons. And then in other cases, they haven't changed, and their job is really to protect and assist neurons. Okay? But we'll come back and we'll talk more about glial cells and neurons later. So the parts of the neuron that are important, they have a cell body. Okay, this is easily distinguished because that's where the nucleus is, that's where all the other organelles would be, that's where the mitochondria would be, that's where the Golgi apparatus, the endoplasmic reticulum, the ribosomes, everything would be in kind of that cell body. Off of that cell body, you're going to have small finger-like projections that go in lots of different directions. These things are called dendrites, and they're going to receive signals, and they're going to have the ability to signal things that are kind of nearby. So if it needs to be one nerve sending a signal to another nerve, okay, that's close, then that signal might move from one to the next. Okay. The axon, on the other hand, is a nerve fiber that's typically longer, okay, than the dendrite. You'll see an example of it, okay? But they're much longer, and so they are an extension of the nerve body that allows the nerve to reach different regions, okay, and interact with the stimuli from a different region, okay? And in some cases, these might be 3 feet long or a meter long, okay? And then organisms that are much larger than us, like a giraffe, we're talking 6, 9 feet. Okay, so 3 meters long. They really have the ability to stretch their signal a long ways and extremely fast. Okay, we'll talk about how fast it is at a later date. Okay, so just an example, neuron with glia cells. The glia cells are the little cells that are outside the neuron, okay, that are helping the neuron by potentially uh, cleaning it, just doing things that would maintain the um, atmosphere or environment around the neuron so it can function as best as possible. And then the potential that some of these may morph into neurons, which we'll get to again. Okay, cell body, nuclei, dendrites, okay, and then an axon. That axon, you can see it's just traveling off in another region, different than the dendrites. The dendrites tend to be short and coming off the cell body, but the axon is much longer. All right, let's dive into muscle tissue real quick. So muscle tissue itself, it contracts when it's stimulated. Okay, and we'll talk about how that works. We'll talk about the actin-myosin interaction later. But it's really important for lots of things. Okay, obviously movement. Okay, but also associated with majority of your organs, all of them, I think, is muscle. Okay, in some form or another. Okay, it allows for organs to flex. It allows for the churning of your stomach. It allows for the forcing of fluid out in certain regions. It allows for the movement of body parts. It allows for really everything. So muscles are super important, and there are different types of muscles. Okay, and one other big thing that we often don't contribute much to muscles because we don't think about it, but muscles generate heat. Okay, and yes, of course, they generate heat when we think about it, we're like, oh yeah, well, if I'm really cold, I shiver. Of course, yes, that's a mechanism to generate heat. But not just that. Okay, just your ability to move, okay, is generating heat. Part of that heat production by moving around, okay, and not shivering necessarily, just moving around, those muscles are going to be firing and generating heat. You lose a lot of heat to the environment around you, okay, and that would maintain heat production so you can maintain homeostasis. Skeletal types are 3 types of muscles. Okay, you have skeletal, cardiac, and smooth. Okay, skeletal muscles, most of the time they are multinucleated. Okay, so they have multiple nuclei inside the cell. They can be extremely long cells. One of the best ways to distinguish them from other types of muscle is the striations. Okay, so they have the strand-like striations, which allow them to contract and then and then expand back out. So the striations are really important. But again, you're going to probably see multiple nuclei within a single cell, and these cells can be fairly long. Okay. Skeletal muscles in general, we classify them as what we call voluntary muscles. Okay, so you have the ability to control these muscles. You have the ability to voluntarily flex these muscles, voluntarily contract these muscles. Okay, versus the other two muscles, which you don't have the ability to do so. So cardiac muscle, involuntary. Okay, again though, they have striations. Okay, so trying to differentiate between cardiac muscle and skeletal muscle can be difficult until you know what to look for with the intercalated disc, which is going to attach one muscle cell to the next. Okay, and so these intercalated discs allows for signals to move from one cell to the next cell extremely fast and in rhythm. Okay? But that involuntary means you don't have conscious control over this. It's under non-conscious control, meaning it's firing at all times based on a nerve impulse that's being sent, an action potential that's being sent. We'll talk more about the AV node and how that action potential is being sent. What's the stimulus look like? What's going on? But just in general, looking at the cardiac muscle, still has striations, but now it's got these larger kind of disc-like units in between cells called intercalated discs. Okay? And that's a good kind of giveaway that you're dealing with cardiac muscle, not skeletal muscle. And the last type of muscle is smooth muscle. It gets its name because there's no striations. Okay? Again, they can be extremely long. Occasionally you could have multi-nuclei in the cell, but most of the time it's single nucleated cells. But again, long, but probably not as long as skeletal muscle. Okay, and there's no striations. These are also involuntary muscles. Okay, so you don't really get to flex these muscles. These are muscles that are associated with things like the churning of your stomach, the intestinal, pushing of material out the intestine. Okay, movement of material around the bladder. Okay, so these muscles tend to involuntary, happen to be the ability to constrict, etc., based on stimuli from within the body. Okay. Tissue growth. Let's talk a little bit about tissue growth. Again, we'll come back to muscles. We'll come back to nerves, and look more in detail at many of those and how they work. Okay. But before we get into, actual types and things like that, let's talk about how these tissues grow. Okay? Growth is normally dividing of cells. We talked about this before. Dividing of cells and adding more cells. Remember, the cells themselves typically are not getting bigger. Right? They're just dividing and you're adding to the number of cells. Okay? Okay, so we call that hyperplasia, is the growth and cell multiplication. Okay? And this occurs during embryonic periods and into child growth. Okay, hyperplasia is occurring. You're growing and multiplying cells, and part of that is differentiation. Okay, and then you have hypertrophy, which is the enlargement of pre-existing cells. Okay, and so some cells, right, like your muscle cells, which are already elongated, okay, you can enlarge them. You can add material to them to make them a larger cell. They can actually grow larger. Fat can do the same. Okay? Nerve cells can do the same. They can grow longer and larger as you increase in size. Okay? Neoplasia is the development of a tumor or a neoplasm. Okay? And that is typically distinguished by uncontrolled cell growth. Okay, so uncontrolled cell growth, the growth of cells are determined by what we call the cell cycle. Okay, so there are things that have to happen in order for a cell to be able to replicate itself, or in order for the cell to be able to grow. Okay? And if they get past all those checkpoints, then they're good to go. But neoplasia means that probably one of those checkpoints is being bypassed and it shouldn't have. Okay, which means that you have uncontrolled cell growth and the tumor develops. Now, these tumors can be benign or malignant. Benign means that that tumor is encapsulated. Okay, and so that tumor, it's there, it was uncontrolled growth, but it becomes encapsulated and it can't spread. Malignant means that it can spread, like little parts, cells will break off and move to a different region, and that tumor will continue in a different region. Okay. All right, so tissue can change types within certain limits. Most of you have probably heard of stem cells. Most of you are probably a little bit familiar with them. But when when sperm and egg come together, they form what we call a zygote. And at that point in time, all cells are embryonic stem cells. Okay? And then as that zygote starts to divide and become more developed, you'll have regions of the body that start to form. And those regions of the body have certain cell types. And so those cells in that region, like say your head, these cells that make up your hair follicles, they become differentiated into hair follicle cells, which means the genes that are within inside that cell turn on the proteins needed to make hair and turn off the proteins that need to make, say, a hormone, or to say to make red blood cells or something like that, or really anything else. It's just these are differentiated. Those genes that are needed to turn on to make cells that can make protein that develops keratin are turned on. Okay, and so we talked a little bit about cartilage and bone, okay, but an example of this differentiation is embryonic mesenchyme cells become cartilage, and then the cartilage, and they also become bone, but then that cartilage ossifies and becomes bone. Okay. Differentiation occurs still today as an adult, okay, but just at a more localized region. Right? And so you have stem cells throughout your body at localized regions, but those stem cells they have limits to what they can be. Okay? They have limits to what they can change into. Okay? Metaplasia is changing one tissue type for another. Okay? This can happen with fluxes of hormones. Okay? So for example, during puberty, the change from cuboidal tissues to stratified tissue occurs, especially in the vagina. Okay. Other things that can happen, again, it doesn't have to just be hormones. It could be stimuli damage from the environment. It can be overexertion in regions, things like that. Just extreme change or extreme environments can change things. For example, people that smoke, not all the time, but lots of times smokers will have pseudostratified epithelium, okay, and it will change over to stratified squamosous epithelium. Okay? And so instead of being pseudostratified and potentially having a longer connection, now it's stratified and you'll get the cells will build up, and that kind of gives you this scar tissue that's formed. Okay, and this can happen over time due to the deterioration of the pseudostratified layer. All right, stem cells. Again, undifferentiated just means they don't have a function, a specific function yet. They have all the genetics. They could technically be whatever they want to be or whatever is needed. Okay? And that's exactly the purpose of a stem cell. Is it's a juvenile cell typically that has the potential to be a few different types. Okay, it depends on where that cell is. Now, there are different types of stem cells. Embryonic stem cells, okay, again, these are cells that occur in the human embryo. They can pretty much be anything. Okay. Totipotent stem cells, okay, very early on, they have unlimited developmental plasticity, meaning it could develop into a nerve cell, it could develop into a muscle cell, it could develop into epithelial cells, anything, right? Some kind of connective tissue, red blood cell, white blood cell, whatever. Okay. Pluripotent stem cells, they have a little bit more limit depending on where they're deposited at. Okay, so they're called embryoblasts. Okay, so it will determine, right, they what type of cell they can differentiate into is determined by where it occurs in the inner cell mass. Okay, and so often you got no accessory organs, so these aren't going to become things like digits or anything like that. And then the ones that we most often are dealing with currently in our life are adult stem cells. Okay? And so this will get at some of the topics that we'll talk about as we progress in class is totipotent stem cells are by far the most valuable. Okay? Especially if you got someone that needs stem cells to maybe replenish, maybe we're trying to help with kidney function or something like that. If you use a totipotent cell and you inject it into that region, it will be kidney cells. Okay. If you use the adult stem cells, you have to get the adult stem cells from that region. Okay, so they're undifferentiated cells, but they're found in the region of where they're going to grow. Some can be multipotent. Okay, they could develop in 2 or more cell lines. Okay, but majority of the time they're unipotent. Okay, one cell type. Okay, so that's like a single cell. There's a single cell that can give rise to sperm. Okay, so this is kind of a sticky issue. Embryonic stem cells, by far the most important and useful for stem cell research, but also stem cell treatments. Okay, adult stem cells are still useful. There's less of them, and the undifferentiated cells still they don't have much of a choice. Can't really develop into anything. That's what I'm getting at. Okay. All right, so let's talk about tissue repair. Some type, maybe some uses for stem cells. Okay. So you can have damaged tissue can be repaired in two main ways. Regeneration, okay, that's the replacement of dead or damaged cells by the same type as before. Okay? And sometimes this occurs just because the material can regrow and can go through mitosis and start developing new material, like your liver, for example. Your liver, you can have cirrhosis of the liver or dead material in your liver, and if you stop what's killing the liver, your liver can replenish itself. Okay. In fact, you can donate over half your liver and your liver will regrow itself. Okay. So there's regeneration. Certain things in your body have the ability to regenerate. Okay. Of course, many of you have cut your finger. Okay. That's a minor skin repair. Regeneration. Okay, the skin cells that are near that cut are stimulated to go through mitosis. They start developing, they close the gap, and you regenerate that skin. Okay. The other way at which this can happen is if it's a deep cut or deep damage, you can have what we call fibrosis, okay, which is where scar tissue is laid down. Okay. And so this is different because it does not restore the function in that region. Okay? It will provide the protection needed, but it doesn't restore function in that region. So regeneration restores function. Okay, so you can regrow big chunks of your liver, function. Okay, but if you have damage, okay, to like your lungs, and you develop fibrosis, that lung, that scarring of the lung, it's no longer functional. Okay, so you're going to have a decrease in your ability to transfer oxygen into the blood in that region. Okay, tissue degeneration and death. Okay, atrophy is the shrinkage of tissue or loss of cell size. Okay, this happens all the time. Okay, aging happens, or lack of use can do this. Okay, and we'll talk about some examples of that as we progress. More importantly, and more often, what's seen and dealt with on a kind of day-to-day basis is necrosis or tissue death. Okay, and there's lots of ways at which this can happen. Infraction, okay, is when blood supply is cut off, the nutrients are cut off, the oxygen's cut off, and waste gets developed or waste gets just accumulated in that region. Okay, and then that tissue will start to die back. Gangrene, also due to insufficient blood supply. Okay, and there's two kind of types of gangrene. So the difference between these two is normally gangrene is associated with an infection. Okay, so you had a bacterial infection, a viral infection, okay, some kind of infection in that region, and then develop gangrene, which you have insufficient blood supply to that tissue type. Okay, so you can have what we call dry gangrene. So it doesn't have a moist layer to it. This happens often in individuals who might be paraplegic and they're in a bed in a wheelchair. They can develop what we call bed sores or pressure sores. Also complications with diabetes. Okay, lack of blood flow to the extremities, to the toes, the fingers. You can get dry gangrene in those regions. Wet gangrene is more liquefaction. Okay, liquefaction of the organs with an infection. Okay, and so this can be a liquefaction of the epidermis. It could be a liquefaction of connective tissue, even muscle tissue, and so that's typically called wet gangrene because there is liquid associated with it. And gas gangrene is when you get bubbles, hydrogen bubbles forming in the tissue. Okay, and so often all these kind of types of gangrene are associated with some type of infection. Sometimes it's purely bacterial, but again, uh, just different types of way that tissues can degenerate and die. So here's just a couple examples. This is dry gangrene. Okay, probably individual had diabetes, and what that often happens is blood flow doesn't make it to the toes, and then the tissue just necrosis and dies. Okay, gas gangrene. Here you can see here, it looks like a blister. It's kind of filling full of hydrogen. Because the tissue's breaking down and gases being formed underneath. Okay. All right, apoptosis. This is natural. This happens all the time, and for good reason. Okay, sometimes your cells get old, and sometimes they need to die. And so apoptosis is programmed cell death. And so not only is that for cells that are kind of out of whack, but it's also when you develop in the womb, your fingers are all fused together, okay, and then apoptosis splits the fingers and the toes. It removes underneath your tongue. Your tongue is tied down to the bottom of your jaw, and apoptosis will remove that little bit of skin flap so the tongue can move and fingers can move individually. Okay, so it's not just single cell, it's regions. It can happen uh, during the development of the fetus, but it can happen later on in adulthood that you're going through apoptosis and normal cell control or cell programmed cell death. And there's a few ways at which this can happen. Okay, it can just be phagocytized by your macrophages, by your large white blood cells, can just phagocytize that cell that's maybe damaged or not doing well, or maybe the cell has went through the checkpoint and it voided, or went through the cell cycle and voided some of the checkpoints, so it gets phagocytized. Lots and lots of cells die via apoptosis. Happens all the time. And so all your cells will have kind of a built-in suicide program. If they are damaged to the point where they can't make a cell that can carry out its normal function or that can replicate, then typically it has a built-in suicide or apoptosis.